Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the bioinformatics. In my previous video, I've told you that how you are going to read the sequences of the DNA in the right way. And in this particular video, I'm going to focus on the two sides of a DNA sequence, which we technically call is the complementarity of the DNA sequence. Now, I was telling you the history as well, how the process of the or the subject of the bioinformatics that started. So, in the same laboratory where Kendrew and Perutz, they were trying to figure out the first 3D structures of a protein. As I've told you that the story of the bioinformatics started with the proteins. So, in the same lab, Watson and Crick in 1953, they elucidated the famous double helical structure of the DNA molecule. And they told us that the DNA molecule consists of two complementarity strands or two complementary strands. So what we means by these two complementary strands? So by complementarity we mean that the thymine on one strand of a DNA that is always going to face an adenine on the other strand of the DNA. Similarly, the guanine on one strand of the DNA that is going to face a cytosine on the other strand of the DNA and vice versa. So if on one strand there is thymine that is going to attach with the adenine, and if there is adenine that is going to attach to the thymine, same will be true for the guanine and the cytosine. So if you look at this, as I've told you in the last video, there are usually two broad classes of the nitrogenous bases. The thymine and the cytosine, they belong to the pyrimidines. The adenine and the guanine, they are going to belong to the family of the purines. So what this simply mean is that a pyrimidine is going to interact with a purine, and again a purine is going to interact with a pyrimidine, but specific purine is going to interact with specific pyrimidine, like the thymine with the adenine and the guanine with the cytosine. Now, why this is important, why the complementarity of the DNA sequence is important. One of the importance is when you know the sequence of nucleotides along one strand, you can automatically deduce the sequence of the other one. This is one of the importance of the complementarity of the DNA strands. For example, if you know the sequence of this particular strand of the DNA, so if there is adenine, you would automatically deduce that on the other strand there would be a thymine. And if you know the sequence of this particular strand, and if you know that there is a thymine, so automatically you will know that the adenine will be present on the other strand of the DNA. Similarly, like guanine with the cytosine, the thymine with the adenine. So this is one of the advantages of the complementarity of the DNA, that if you know the sequence of one strand of the DNA, you can deduce the sequence of the other strand of the DNA. Another important thing is about the complementarity of the DNA is living organisms like the human beings, when they reproduce, they are going to replicate their DNA. What I mean by replication is that they are going to duplicate their DNA. So in nature, what happens is that the DNA strands, they are going to separate from each other and make two complementary ones. What I mean by that is that if this is the double-stranded DNA, the red is one strand, the blue is the other strand. So during the replication, these two strands, they are going to separate from each other, and utilizing the machinery of the DNA replication, they are going to make the new strand from themselves. That means that from one copy of the DNA, you are going to make two copies of the DNA, and that is following the complementarity of the DNA. Now, when we talk about the sequences of the DNA, of course, in bioinformatics, you are interested in determining the sequences of the DNA and hence the impact of the DNA sequences on different parameters. So the IUPAC, they have mentioned one letter code for the DNA sequence, and that is the A is for the adenine. Of course, we belong to the category of the purine. For cytosine, the one letter code is C. For guanine, it's G. For thymine, this is T. So if you are sequencing, if you are having a sequence of the DNA, and if you find A, that would refer to adenine, C to cytosine, G to guanine, T to thymine. If there is an N in the sequence of the DNA that you have got after the sequences, that means it can be any nucleotide or it can be any base. If there is an R letter in your the sequence that you got from your company, that can refers to a purine, and the purine can be adenine or it can be guanine. Similarly, for pyrimidines, if there is a Y, so Y can be a cytosine or it can be a thymine. And if you are having these things, that means that there is a gap in the sequence of the DNA. So this is how the IUPAC they are going to use the one letter code for the DNA sequences. So in the next video, I'm going to talk about the palindromes in the DNA sequence. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.